Hello and welcome to Physiology with Shakthi. Now, in the previous episode, we had done with neural regulation of respiration. So, if you haven't checked it out already, please go and do that. Now, in today's episode, we're going to understand how chemical regulation of respiration works. When we talk about chemical regulation of respiration, we have to understand that it detects the chemical composition of the blood. So what exactly is the composition of blood that it regulates? That is what we need to understand right now. So we got the oxygen levels, we got the carbon dioxide levels, and we have the concentration of H plus. Now, how is this detected? Which component of this is detected? We got the partial pressure of oxygen. So, in the dissolved state, it exerts a partial pressure. So, that is what is detected. And then we got the partial pressure of carbon dioxide. And H plus concentration is given by your pH values. So these are the basic components that are regulated, that are monitored and regulated. So that is where your chemical regulation takes place. So that takes place to two set of receptors. They are chemoreceptors. So these chemoreceptors are present peripherally in your body, and there is a central chemoreceptors in the central nervous system. So we got the central chemoreceptors and we got the peripheral chemoreceptors. So now we'll try to understand how they work and how different they are. So when we talk about your peripheral chemoreceptors, let's take a look at your aorta first. Now if we take a look at the aorta, we got the left subclavian coming out from here. We got the common carotid coming on the left side, and on the right side things are slightly different. It comes from your brachiocephalic trunk, from where your right subclavian comes, and then followed by your common carotid. So we got the common carotid on either side. Now this common carotid divides into your internal as well as the external carotid artery. We got the external carotid artery, and laterally to it we got the internal carotid artery. So we got the common carotid artery, we got the external carotid artery, we got the internal carotid artery. Now this is where people get confused a bit. We got two sets of receptors. We got the central as well as the peripheral chemoreceptors, and this is your peripheral chemoreceptors. But people get confused between which is a chemoreceptor and which is a baroreceptor. So that is another thing that you need to understand right now. So we got sinus as well as bodies. Carotid sinus, carotic bodies. Aortic sinus, aortic bodies. So which is for chemoreception, which is for baroreceptor. So that is what we need to understand. So baroreceptors detect pressure and the volume. This pressure and the volume is what baroreceptors detect. So we got a sinus over here. This part over here that we see, that internal carotid just begins, this initial part. So this part has terminal ramifications of branch knobby chains. So this detects some things. So now question is the sinus over here, whether it detects pressure or your chemical signals. So when we got a blood vessel like this, what is blood pressure? It is the lateral pressure exerted on the vessel wall. This lateral pressure is detected by this sinus, this carotid sinus, where terminal ramifications of nerve endings are present, detects your pressure and the volume. So that is not your chemical regulator. That is not the receptor for chemical regulation, nor the peripheral chemoreceptors. That is for blood pressure. Apart from this, over here we got a particular body like this. Over here too we got a body like that. So it is a closed chamber. It's not closed. So this is a separate chamber, a sort of a box opening out like this. And this box has certain receptors. So blood which is flowing goes in, comes out. So this is richly innervated to detect chemical composition of the blood. So that body is your peripheral chemoreceptor. So you don't have to get confused now. The sinus is for baroreception. Your body is for chemoreception, peripheral chemoreception. So we got the carotid bodies, we got the aortic bodies. So carotid as well as aortic bodies are present. So these have one of the highest blood flow rate when you take a square centimeter area. So when you take a square centimeter area, you realize that this has one of the highest blood flow in the body. The small little carotid body has one of the highest blood flow in the body, so that it can easily detect small changes in chemical composition. So that is your peripheral chemoreceptors, and these chemoreceptors are innervated. So we got your carotid. So from the carotid, it's more above, that is your 9th cranial nerve. And from here we got the 10th cranial nerve. So from the aortic bodies we got the 10th cranial nerve. From the carotid bodies we got the 9th cranial nerve. So you understood your peripheral chemoreceptors now. These peripheral chemoreceptors are carotid and aortic bodies. Now apart from this, we got your central chemoreceptors. When we talk about the brain, we got the medulla, we got the pons. So in this medulla, we know that there are ventrolateral areas like this. Just know that there are actually 3 sets of defined areas that have been discovered, and based on who discovered that, different names have been given. But at your level, I don't think you need to understand those right now. Just know that in the ventrolateral region, there are set of areas which can detect the chemical composition. What chemical composition and how it detects is what we need to understand. So that is your central chemoreceptor. So that detects chemical composition. Now what is detected by what is what you need to understand. We got the peripheral chemoreceptors. In the periphery, oxygenation, when it drops, when the PO2 drops, that is specifically detected by your peripheral chemoreceptor. It's not like PCO2 and pH is not detected. They are also detected. But it is said that your peripheral chemoreceptors are more sensitive, are more important for detecting PO2 changes. And in a similar fashion, we got either PCO2 or pH now, which will determine your central chemoreceptors. Now we need to understand this certain question. Why have we evolved in such a way that instead of looking at PO2, we are looking at something else like the PCO2 or the pH? Why is there a need for that? So take a look at this diagram, the oxygen hemoglobin dissociation curve. Now if you take a look at this diagram, if you look at the drop in PO2 from 100 to 90 to 40, so from the alveolar level to the blood vessels to finally when it reaches the tissue level. So at that point you see that the PO2 is dropping from your 97 to 40 millimeter of mercury. So when this drops, you can notice that the hemoglobin is less saturated. So from the initial value, the hemoglobin saturation has gone down to 75%. So what does it mean? By the time it reaches your tissue and the PO2 falls over there, accordingly your hemoglobin dissociation is changed. So when the body in the periphery needs oxygen and when the PO2 falls, accordingly your hemoglobin adjusts in such a mechanism so that the oxygen is released from hemoglobin. So more fall in PO2, more oxygen is released. So your body has a very clever mechanism to tightly regulate how much oxygen is received at the tissue level, because lack of tissue level oxygen is what results in hypoxia. So to prevent such a hypoxia, what the body does is it regulates the PO2 by dissociating more oxygen from the hemoglobin. That is basically what you understand from your dissociation curve, the oxygen hemoglobin dissociation curve. So when something like oxygen, which is that tightly regulated to make sure that at the tissue level it is not that affected even if small changes in PO2 are present, will that be a good mechanism to be a central and the most important mechanism to regulate chemical composition? No. Because it is tightly regulated. Even if small fall in PO2 is there, your body has mechanisms to regulate it by itself without using your chemical regulators. So that wouldn't be an ideal and a very powerful mechanism to regulate it. So that is why we evolved in such a way that PO2 is detected predominantly by your peripheral chemoreceptors. On the other hand, your central chemoreceptors is going to derive another mechanism to detect chemical composition. So what does it do? We got something called the blood-brain barrier over here. Do you think H plus ion concentrations cross the blood-brain barrier? It does not. Water-soluble things like H plus do not cross the blood-brain barrier. So things like H plus do not cross the blood-brain barrier to reach inside. So what is remaining? We got the PCO2. So the PCO2. So the carbon dioxide goes inside and it combines with water. It forms the H2CO3, the carbonic acid, which forms the H plus and HCO3 minus. Now this mechanism is in such a way that it is a reversible reaction. So more PCO2 is there, more CO2 is there, more H plus is generated. If this is lesser, it goes back. So it's kind of like that. So when more PCO2 is there, more CO2 is there, ultimately more H plus is generated. And more H plus is generated means that is ultimately what is sensed by your chemical regulators. So these receptors, the central chemoreceptors, actually detect H plus ions. So this is where I think questions come. Even if you know this topic, they might confuse you if you don't read the question properly. What is detected in the blood? PCO2. Central chemoreceptors detects PCO2 in the blood. But what does the central chemoreceptor actually physically detect? The H plus concentration, not carbon dioxide. So central chemoreceptors detect the H plus concentration in the brain. But that is synonymous actually to your PCO2 levels in the blood. But does it detect pH, the H plus concentration in the blood? No. So it detects the H plus concentration in the brain, but not in the blood. So these are various things by which they can confuse you. So once you understood this, you need to understand something else. When you talk about central chemoreceptor and peripheral chemoreceptors are there peripherally outside the brain. While this whole process will take a little time. It's going to go to the brain, cross the blood-brain barrier, form CO2, H2O2, carbonic acid, then split into H plus and HCO3 minus. So that is a issue over here. So this peripheral chemoreceptors has been kept there so that is a very fast mechanism which is present. This is 5 times faster than your central chemoreceptors. Compared to central chemoreceptors, your peripheral chemoreceptors are 5 times faster. But the central chemoreceptors are more powerful. So they are up to 7 times more powerful. So they're more potent, 7 times more potent than your peripheral chemoreceptors. So we got the PO2. It also detects PCO2 and pH. Very different mechanism. We'll come to it a little later. And your central chemoreceptors predominantly measures your PCO2 levels in the blood. Are we clear with this? So now that you understood this, we'll go on to understand the structure of your peripheral chemoreceptors. Questions are asked being based on that. So when we talk about the peripheral chemoreceptors, we got 2 types of cells. I told you these go in, blood comes in, and that is detected by a group of cells over here. And that is carried finally by these cranial nerves. So what exactly is happening there? We got 2 types of cells over here. We got one major cell, which is a type 1 cell. So type 1 glomus cell. And we got the type 2 glomus cell. These type 2 glomus cells are sustentacular cells, a supporting cells. While your type 1 is a major glomus cell. Again, the next question. So these questions are asked for MCQs. So your type 1 glomus cell is what actually works for detecting this. That is the main chemoreceptor, peripheral chemoreceptor. While your type 2 cell is supporting cell. Now we had seen these cranial nerves coming out from this. So that is your cranial nerve over here. Efferent comes out and goes. Where does it go? It goes from here and finally reaches parts of your nucleus tractus solitarius. Once it reaches the nucleus tractus solitarius, from there it is now going to go to your central pattern generators. So there are 2 things happening over here. We got your peripheral chemoreceptors which reach your nucleus tractus solitarius. And we got your central chemoreceptors in the ventrolateral region of the medulla. Now where are the central pattern generators? They are present over here. We got the dorsal respiratory group. We got the ventral respiratory group like this. Right? So these are your central pattern generators where impulses, respiratory neurons are present. So this impulse that reaches from here will now go and regulate this. Impulses coming from your central chemoreceptors will again go and regulate this. Thus, they can inhibit or increase your respiratory neuron discharge. So what is your respiratory neuron discharge? That is the inspiratory RAM signal that is present. You already seen that. So we see this inspiratory RAM signal. Now, if it detects that PO2 is less or PCO2 is more, what does it mean? PO2 is less. PO2 is less means you need to ventilate more so that more oxygen comes in. PCO2 is high. PCO2 is high as in there is more carbon dioxide, so we need to breathe faster to wash out that carbon dioxide because it can be a potential carbonic acid and it can change the pH, it can reduce the pH. Or if the pH is less, that means you already have a lot of H plus present, so you don't want a more potential PCO2 that can result in more acid production. So again you ventilate and wash it out. So when PO2 is less or PCO2 is more, or when the pH is less or H plus concentration is more, we try to increase your rate of respiration. You need to breathe faster. So what it does is, it does 2 things. This regulates the respiratory neurons by changing 2 separate things. It can increase the rate. It can increase the rate of rise. So the rate of rise of ramp signal. So the slope can be changed and the rate is increased, as well as it can increase the depth of respiration as well. You can already, if the rate is higher and it goes this normal speed, the depth is already increased. So with that it can further also change it. So two things it can do. It can control the rate, it can control the depth. So two things that it can do. So ultimately, when peripheral chemoreceptors and central chemoreceptors act, they do two things. It becomes a rapid process, rapid rise, and there is also increase in the depth. So the rate of inspiration, the depth of inspiration is increased. That is what your chemoreceptors do. You consider a situation where PO2 is there, lot of PO2 is there, no signals from here arise. PCO2 is less. So no signals arise to stimulate that. So when these signals are not present, what is it going to do? They are going to inhibit this so that the rate of rise is slower and it can be shorter also. Understood? So that is what your chemical receptors do.